Assalamu alaikum students. Hope you all are fine. This is Afsa Hassan. Today we have to discuss chapter number 6 Enzymes from your biology book. Open page number 109. The topics that we have to discuss are factors affecting the rate of enzyme action, mechanism of enzyme action, and specificity of enzymes. There are different factors that affect the rate of enzyme action. Number 1, temperature. 2, substrate concentration. And 3, pH. We will discuss all these factors in detail. First one is temperature. Come to page number 110. As we have discussed earlier, that enzymes are proteins which speed up the chemical reaction. Every enzyme works at its optimum temperature. Optimum temperature is the temperature at which the enzyme works at its maximum rate at a specific temperature. That specific temperature is optimum temperature. When the temperature is raised to a certain limit, heat adds in the activation energy. Activation energy is the minimum amount of energy that is required to start a chemical reaction. When the temperature is raised, heat energy is added in the activation energy and kinetic energy is provided to the reaction. When kinetic energy is provided to the reactions are accelerated. When the temperature is raised well above the optimum temperature, heat energy increases the vibrations of atoms of enzymes and the globular structure of enzyme is lost. This is known as denaturation of enzyme. When denaturation of enzyme takes place, it results in a rapid decrease in rate of enzyme action, or it may be completely blocked. The optimum temperature for the maximum working speed of human enzyme is 37 degrees Celsius. Means if the human enzymes are provided the temperature less than 37 degrees Celsius, the reaction rate will slow down. But if the human enzyme is provided temperature more than 37 degrees Celsius, reactions are blocked completely or it will rapidly decrease. On the other hand, birds have higher body temperature than mammals. They have the specific temperature that is 40 degrees Celsius. It means that the enzymes that work in the body of birds works at its maximum rate, and that is 40 degrees Celsius. If the temperature is less than 40 degrees Celsius, the reaction will slow down. Or if it is provided with temperature more than 40 degrees Celsius, reaction will be blocked completely. Look at the graph. As the temperature increases, so does the rate of enzyme activity. An optimum activity is reached at the enzyme's optimum temperature. Optimum temperature of human enzymes is 37 degrees Celsius. A continued increase in temperature results in sharp decrease in activity of an enzyme as the enzyme's active sites are denatured. When we have to show the graph, we have to show on the x-axis rate of enzyme catalyzed reaction, and on the y-axis we have to show temperature. Temperature at which the human enzymes work at their maximum rate is 37 degrees Celsius. If they are provided with temperature less than 37 degrees, reaction rate will slow down. But if they are provided with temperature more than 37 degrees, it will result in denaturation of enzymes. Then come to page number 110. The second factor that affect the rate of enzyme action is substrate concentration. If enzyme molecules are available in a reaction, increase in substrate concentration increases the rate of reaction. If enzyme concentration is kept constant and the amount of substrate is increased, a point is reached where any further increase in substrate does not increase the rate of reaction anymore. How it takes place? For example, there is a reaction in which the concentration means amount of enzyme is kept constant, but the amount of substrate is increased. The reaction will speed up until all the active sites of enzymes will be occupied. Once all the active sites have been occupied, then any increase in the amount of substrate will not increase the reaction rate because they will not find any empty active site to bind with. So the reaction will not increase. It will remain constant. Look at the graph. On the y-axis, we have to show substrate concentration, and on the x-axis, we have to show rate of enzyme catalyzed reaction. Increasing substrate concentration also increases the rate of reaction to a certain point. Once all the enzymes have bound, any increase in substrate concentration will have no effect on the rate of reaction, as the available enzymes will be saturated and working at their maximum rate. Third important factor that affect the rate of enzyme action is pH. But before moving towards pH, here we have pH scale that is necessary to understand. What is pH scale? pH scale measures how acidic or alkaline a substance is. pH scale ranges from 0 to 14. A pH of 7 is neutral. Water is neutral because it has pH 7. pH less than 7 is acidic. Those substances which have pH less than 7 are acidic. pH more than 7 is basic. Understand? pH scale measures how acidic or basic a substance is. Like temperature, all enzymes work at the maximum rate at a narrow range of pH called as the optimum pH. All the enzymes that work in human body have a specific pH at which they perform their action. If the change in pH takes place, it will result in retardation in enzyme activity or blocks it completely. For example, pepsin is an enzyme that works in the stomach cavity. Pepsin enzyme work only in acidic medium. Acidic medium, low pH. As I have said that pepsin works in stomach cavity, pepsin has pH 2. Trypsin is active in alkaline medium. Trypsin is an enzyme that works in small intestine and it has high pH. Its pH is 8. pH change can affect the ionization of amino acids at the active site. Look at the graph. The most favorable pH value, the point where the enzyme is most active, is known as the optimum pH. This is the optimum pH. For example, the pepsin enzyme has optimum pH 2, so it will work at its maximum rate at 2 pH. While the trypsin enzyme that works in small intestine has pH 8, so it will work at pH 8 at its maximum rate. Extremely high or low pH values generally results in complete loss of activity for most enzymes. The second topic that we have to discuss is mechanism of enzyme action, that is on page number 111. How do the enzymes perform their activity? When enzyme attach with a substrate, a temporary enzyme substrate complex is formed. Enzymes speed up the reaction and substrate is converted into products. After this complex breaks, enzymes and products are released. When the enzymes perform any chemical reaction, they do not change, only products are released. They will remain same throughout the reaction. Enzyme plus substrate. A temporary enzyme substrate complex is formed. Look at the diagram. This is the enzyme and this is substrate. When they react with each other, a temporary complex is formed that is called as enzyme substrate complex. After that, enzymes will remain the same and products are released. Lock and key model was introduced in 1894 by Emil Fischer, who was a German chemist. According to the lock and key model, both enzyme and substrate possesses a specific shape. The shape of substrate is just like the shape of the active sites. That fit exactly into one another. This model explains enzyme specificity. Look at the diagram. Here we have shown substrates and these are the active sites of the enzymes. The substrates have the same specific shape like the active sites of enzymes has. Enzyme substrate complex will be formed because the enzymes and the substrates have specific shapes that they fit exactly into each other. When the reaction will proceed, enzymes will remain the same and products are produced. Induced fit model was proposed by Daniel Koshland in 1958. This induced fit model was actually a modification to lock and key model. He said that according to the model, the active site is not a rigid structure, that the active site is not a hard structure. It is molded into the required shape to perform its function. Means that the active sites are not rigid, they can change their shape according to the shape of the substrate. Induced fit model is more acceptable than lock and key model of enzyme action. Look at the diagram. This is the shape of substrate and this is the shape of active sites of enzyme. They both are different. According to induced fit model, these active sites are not so rigid. They can change, they can mold their shape according to the shape of substrate. Look at the enzyme substrate complex that the active site has changed its shape according to the substrate and they fit into each other. Specificity of enzymes. There are over 2000 known enzymes and each enzyme is involved in one specific chemical reaction. Enzymes are also substrate specific. Specificity of enzyme is determined by the shapes of their active sites that fit with the substrates. Active sites possesses specific geometric shapes that fit with specific substrates. The geometric shape of active site of enzyme determines its specificity for substrate. For example, protease is an enzyme that works on proteins. Its function is to break down peptide bonds in proteins. Amylase enzyme works on starch. Lipase enzyme acts on lipids and it breaks them into fatty acids and glycerol. These all enzymes are specific in their action, means that protease enzyme will not work on starch as well as amylase enzyme will not work on lipids. They all are specific in their action and in their chemical reactions. Look at the diagram. As I have said that enzymes are very specific and work with their substrates. This is the substrate and it will fit into the active site of the same shape. This substrate is specific for this enzyme. It will not bind with the other enzyme because it has different shape of active site. Same is the case here. This substrate will bind with this specific enzyme. This substrate will bind with the enzyme which has the same shape, same active site. Active site is the site where the substrates bind. Here is another example. This is the enzyme type 1 and specific substrates will bound with the enzyme of type 1. Here we have enzyme type 2. Look at the active site that is different from the type 1. So the specific substrate for enzyme 2 is this one. Look at the active site of enzyme type 3. So it has its own specific substrate. Now come to review questions. On page number 116, understanding the concepts. Question number 3. In a range of 0 degrees Celsius to 35 degrees Celsius, the rate of reaction of an enzyme is proportional to temperature. Above 35 degrees Celsius and below 0 degrees Celsius, enzyme activity slows down and eventually stops. Explain why. So the answer is increase in temperature will speed up the rate of enzyme catalyzed reaction, but only to a certain limit. Every enzyme works at its maximum temperature. If temperature is raised above to a certain limit, heat energy adds in the activation energy and provides kinetic energy to the reaction. So, reactions are accelerated. When the temperature is raised well above the optimum temperature, the heat energy increases the vibrations of atoms of enzymes and the globular structure of enzyme is lost, which results in denaturation of enzymes. Rate of enzyme action decreases or it may be blocked completely. Conclusion. So above 30 degrees, 35 degrees Celsius or below 0 degrees Celsius, enzyme activities slow down and eventually stops. Question number 4. How does pH affect enzyme activity? Answer is on page number 111. We have discussed about pH that like temperature, also the enzymes have their own specific pH at which that they perform their activity. That pH is called as the optimum pH. For example, pepsin is an enzyme that works in stomach cavity and it works in acidic medium, while trypsin is enzyme that works in small intestine and it works in alkaline medium. Question number 5. What characteristic of enzymes makes them specific for substrate? Answer is on page number 110. That is substrate concentration. For example, if we have 10 enzymes and 20 substrates, reaction will speed up until 10 substrates join with the active sites of enzymes, but the remaining 10 substrates will not increase the rate of reaction because they will not find any active site to bind with. The reaction rate will remain constant. Question number 6. Briefly describe the factors that affect the activity of enzymes. Answer is on page number 109, 110, 111. We have discussed all the three factors in detail, that is temperature, substrate concentration and pH. Question number 7. Describe the lock and key mechanism of enzyme action. Answer is on page number 112. Thank you so much.